Good afternoon. My name is Michael Maguire. I'm CEO of Avectas. I'm delighted to be speaking virtually at the Meeting on the Mesa conference. I'd like to, during the course of my presentation today, to update you on considerable progress since we all met in person in Carlsbad last year. And I'm hoping that the fact that we're listening virtually and watching virtually won't in any way diminish the communication of a great deal of progress. So just to give an overview on Avectas, Avectas was founded in 2012 to develop a best-in-class non-viral cell engineering platform. We've an interdisciplinary team of 30, supported by a very experienced board of directors and a world-class scientific advisory board. In terms of commercial activity, we closed a Series C round of $20 million in the first half of 2020. On top, you may remember, of a $10 million Series B round in mid-2019, and bringing our total funding to date to just over $40 million. We have active collaborations with in process development and manufacturing with CCRM in Toronto, with Catapult in the UK, the Karolinska Institute, with UC Davis, and various other undisclosed cell and gene therapy businesses. And in addition to that, we are excited about having signed a licensing and development agreement with Ankh Therapeutics, and we have a preclinical program with Vicelix. I'll speak to both of those during the course of the presentation. So I'd like to focus during this talk on the opportunities for more complex cell engineering. I'd like to speak to our Solupore platform and to deep dive a little bit more on the partnerships and collaborations which we have in progress. So to scene set a little bit, the two main drivers of the cell and gene therapy market over the next 3 to 5 years are likely to be the emergence and maturing of allogeneic approaches, and secondly, the ability to address solid mass tumors. So we've already seen the first generation of single modification CARs go all the way to the market. What's following on that? That's what we're interested in. So in addressing particularly solid mass tumors, we can see on the slide here how hostile the solid mass environment is for a modified CAR T. So that T cell or CAR T cell needs to be as functionally fit as it possibly can be to operate within that environment. And this is where our Solupore platform and our technology wins out. We can affect complex engineering, resulting in highly functional cells which will meet the needs of the industry in this respect for both solid mass tumors and for allogeneic approaches. So to speak about our Solupore platform, Solupore is a reversible permeabilization technology. It comprises a membrane, a delivery solution in which the cargo or cargoes of interest are mixed, and then a range of devices in which to bring that reagent together with a population of cells. We have the Solupore research tool, which is the small device in green on the left-hand side. And this is probably people's first introduction to Solupore, where they can adopt the technology. This is a straight line scale up to the clinical single-use technology, SUT. So this is our clinical offering. It's single-use, it's clinically aligned. It will address between 10 to the 7 and 10 to the 8 cells in a single transfection. We can also batch, and we are very well progressed in terms of our device development pathway, our reagent development pathway, and the regulatory pathways associated with both of those. And lastly, we have in parallel a continuous flow-through system to address higher cell volumes for allogeneic therapies. These embodiments map approximately across the bottom to research scale. The SUT will address autologous scale therapies, and the flow-through systems allogeneic therapies. So to speak about the Solupore process again, it's a minimally perturbating process for cells, but it's effective at engineering them. So we introduce the cargo or cargoes of choice into the delivery solution. This is put into the device here in step 2, at which the cells are introduced to the chemistry. In step 3, the cargo and permeabilization chemistry are introduced to the cells and are transported into the cell. Cellular uptake takes place then, and the cells express the modification which has been delivered to them, and the cells lastly are recovered from the device. So it's a simple, elegant, it's a gentle technology, and it results in cells with greater functionality. So we have spent much of our time focused on T cells, human primary T cells and NK cells. And in terms of cargoes, we've been delivering both model cargoes and gene editing tools to those populations. In all cases, the resulting cells are highly viable. The performance, the efficiency is excellent, and of note in this data here, we can transfect naive cells with some efficiency as well. For the complex manufacturing cases that we spoke to, namely for solid mass tumors or for allogeneic applications, we wanted to demonstrate that we could deliver dual cargo, so delivering 2 cargoes together. So here we deliver a CAR, an mRNA CAR and GFP, and we can see the resulting population of cells. 70% of them are double positive, and also the viability of that population is extremely high. There are also settings where you may not want to deliver all of the cargoes at once. You may want to sequence it out over hours or days, and this can be achieved on the same population with Solupore without a negative impact on the viability of the population. So here we performed an RMP edit of the track locus on day 0 and delivered a CAR mRNA on day 2, and we can see the resulting population of cells has the desired phenotype, 60% of them, and high viability is again retained. We've used the term gentle technology, and I wanted to put some numbers against that. And in this data here, we can see in a comparison with nucleofection, at 2 points, a 6-hour time point and a 24-hour time point, we looked at a panel of immune genes and looked for up and down regulation as a result of the process. And we can see that the nucleofection process, marked in red and green here, there's significant up and down regulation of immune genes of interest within the cells. However, in the Solupore technology, there is much less perturbation, and this situation is the same at 24 hours. And these cells or these pathways which are being perturbed are the pathways of interest in immuno-oncology: activation, metabolism, exhaustion, and so forth, and will certainly be important in the performance in solid mass applications. We can also see that proliferation is maintained in cells which are engineered with Solupore technology. In this piece of data here, we show equivalent proliferation with untreated, whereas nucleofacted cells lag somewhat in their proliferation. We go on to show in this data here about the functionality, so demonstrating the high functionality of the cells. So the Solupore cells and the nucleofection cells are both engineered to express CD19 CAR here, and they're put into an excelligence assay. And although the nucleofection cells express slightly higher amount of CD19, we can see that their function within the assay is at different, at all ratios of effector to target cells is equivalent and in fact better than the nucleofected cells. So this is exciting data validating out the platform. Lastly, in terms of data, we administered Solupore CD19 CAR cells in this animal study. It was performed at Jackson Labs, and a very exciting piece of data I can share with you on day 15 here at the highest dose of Solupore cells, 4 by 10 to the 6. On the panel highlighted in green, we have 3 mice with total tumor clearance as a result of the CD19 CAR T cells administered. In comparison, the same dosage nuclear-affected cells, you do not see the same tumor clearance. So exciting in vivo data there. So I'm going to switch track to speaking to how we're applying this technology in the context of partners. So our key lead preclinical product pipeline partner is Ankh Therapeutics. We're developing a CD19 CAR NK with trail variant, and we're excited about the prospects for that product. We also have a preclinical collaboration with Vicelex in Florida around their YM proliferation enhancement technology. We're excited about the prospects for both of those products. In this slide, I'm just diving a little deeper on the Ankh program. I guess what is key here is that we have licensed rights to the Ankh therapeutic. They will develop it to phase 1, and we expect to initiate a clinical trial within 18 to 24 months. This will validate out the Solupore platform and will constitute a first in man for the platform. In terms of other collaborations and partnerships, we're keenly interested in manufacturing and process development, and we've deepened our relationship announced during the year with CCRM, with UC Davis in California, with the Karolinska Institute, and we have a number of other undisclosed collaborations. So in summary, Solupore is a next-generation cell engineering platform. Next-generation therapeutic products will require complex engineering whilst maintaining cell health. Platforms will need to address key development and manufacturing considerations, and Solupore as a platform achieves the key performance parameters required to constitute such a platform. We've 80 to 85% transfection efficiency in human primary T and NK cells at excellent viability. We deliver a broad range of cargoes, including RNA, RNP, proteins, and combinations. We're gene editing in T and NK cells using multiple gene editing platforms, including CRISPR. We've excellent post-engineering proliferation and demonstrated in vitro and in vivo functionality of engineered CAR T cells. So realizing a platform, a new platform like Solupore, will enable the full therapeutic potential of the solid mass and allogeneic therapies that we spoke about to be conferred on patients, and that gives us a lot to be excited about in the year ahead at Avectas. So thank you very much indeed. I would be delighted to field any inquiries or questions about the presentation. And lastly, the photograph here is back when we could travel, myself and my colleague Gillian Hendy at the ASH meeting last year. So we were putting those little butterflies for cancer on the board there. So we're hoping that we'll see you all again in person, perhaps in Carlsbad next year. But in the meantime, thank you.